Very good afternoon, all of you. Welcome to the EFDP. Hello. Hello.
We can listen to you. Please continue, ma'am.
Okay. Okay. Thank you, sir. Good afternoon, everyone. Welcome to EFDP workshop on waste to bioenergy. Let's start with the afternoon session. Our first speaker of this session is Dr. Piyush Parkhe. Dr. Parkhe is presently working as assistant professor in Amity Institute of Biotechnology, Chhattisgarh. He did his MTech in biotechnology with gold medal from Dr. D.Y. Patil Vidyapeeth in 2009. He completed his PhD in biotechnology from National Institute of Technology, Raipur on DST Inspire Fellowship in 2015. He received Sub-National Postdoctoral Fellowship in 2017 and worked as National Postdoc Fellow in the lab of Dr. S. Venkatamohan. His research interests includes lignocellulosic bioprocessing, alternative fuels, and sustainable energy. He has published 11 peer-reviewed research articles and 5 book chapters. Thank you so much, sir. Thank you for your presence. And now over to you. We are looking forward for your valuable words. I think, wait, sir, just.
Okay. Am I audible?
Yes, sir. Now you are audible.
Anupriya, thank you very much for the generous introduction. And a very good afternoon to everyone. First up, I would like to say thanks and give my I would like to say thanks to the organizers, Dr. Abhilasha, Deepak Jadhav, and Dr. Pandit, who have allowed me to speak on this topic, and to all the organizers, to the whole organizing team on behalf of the Sharda University, who have taken tremendous effort in organizing this EFDP, because these have been unprecedented times of the pandemic, and it really takes in much effort to organize such an FDP. So a big congratulations to all of you, and thank you very much. I hope all of us are keeping safe in these times. So before I begin my talk, I would just like to say that over these 4-5 days of the FDP, we have listened to a number of eminent speakers and scientists who have talked about how to produce energy through waste material like biofuels, production of biohydrogen, methane. They have talked about microbial fuel cells, microbial electrolytic cells. So there have been much discussed and much information has already been disseminated about this topic. So what I was thinking and what I believe is I'll not be talking much about what research I have done. I'll not be talking about actually what I'm doing right now, but I believe that most of the members of this faculty development program per se are the young faculties or the postdocs or the PhD scholars who would be starting their career in a short while from now. So I think that I would be talking about something that is very hot topic of now, and from where I can give the idea of what can be the new thing that can be taken. So I decided about talking about lignin valorization. I would share my PPT. Please let me know if it is visible to you guys.
Yes.
You see? Can you see my presentation?
Yes, sir. Yes, sir.
Yes, sir.
So, as I said, I'll be talking about lignin valorization. Before that, I would just like to give you a brief background about myself, although Kanupriya has given it. This is the place where I work, Amity University, Chhattisgarh. The Chhattisgarh campus per se came up in 2014. It was established by Ratanan Balved Education Foundation, and this building is, this campus is situated at the Raipur Baloda Bazar Road in Raipur. And at the Amity Institute of Biotechnology, we are running UG, PG, as well as research courses. So I am an assistant professor working in here. Before going into understanding lignin valorization, we need to know why lignin? Why do we need to talk about lignin? So lignin, as most of you might know, it is the second most abundant natural renewable polymer which is available on Earth, obviously after cellulose. So the abundance of this lignocellulosic biomass globally has emerged as a solution for the ever-increasing fuel demands and the chemical demands. If you take a number, approximately 150 tons of lignin is annually produced as agricultural waste. So, and this lignin is made up of aromatic polymers. So what happens is this aromatic nature of lignin gives it a greater vitality to be converted into various components. So as you can see, there are a number of keywords with which you can associate lignin to. It's a component of greener chemistry. It gives you a sustainable energy option. The biorefining, it gives you a solution towards climate change. And whatever human impact on environment is, it gives you a technology which is environmentally benign. It gives you a solution for production of chemicals and fuels which are otherwise produced from in industrial processes which are energy intensive. And it gives you a plethora of value-added products. So that is the reason why lignin is such an important product which can be utilized. So when we talk about development, if we develop a technology which can effectively recover and convert this lignin into various value-added products, and such a method is called as lignin biorefinery or lignin valorization. So there has been a tremendous work which has been done in the past decade, as you can see, over the last 10 years, there has been a significant increase in the number of papers, and the last column shows what have been published till this year. So this increase shows how important it is to understand this process and how important it is to get value out of this biopolymer. So now, what exactly is lignin? I don't think that there is much that I need to tell, but still, lignin, as we all know, it is localized within the plant cells and it occurs with cellulose, and it accounts for approximately 50 to 30% of the weight of lignocellulosic biomass. I'll show you with the help of pointer. As you can see, inside the plant cell wall, the lignin, it occurs along with the other two components, which are cellulose and hemicellulose. So it fills the spaces in the cell wall between cellulose and hemicellulose, and its main function is to provide vascular support, mostly in vascular and support tissues such as in xylem or vessel elements or scleroid cells. And this cross-linking provides the mechanical strength to cell wall and as such to the whole plant, right? So this is the representative of lignin structure. As you can see, it's chemically is, you can call lignin as a cross-linking phenolic polymer. What is the structure of lignin per se and the individual monomers? I'll discuss it in a short while, but before that, let us see why specifically there is such a great, such a developed interest in lignocellulosic biorefinery. So this flow chart you can see summarizes the steps of the feedstock, lignocellulosic feedstock biorefinery. As we saw in previous slide, that there are three major components which are present in lignocellulose. These are lignin, hemicellulose, and cellulose. So all these three components can be effectively converted into value-added products, into commodity products. And that is the reason why, particularly, lignocellulose as a substrate has attracted such attention in the recent past. So you can get biofuels, you can get fine chemicals, you can get polymers, other materials, pharmaceutical grade compounds, you can produce heat, other ways of energy, extractives. either these compounds can be directly used, as you can see, cellulose or hemicellulose or lignin can be directly burned to get heat and power, or they can be processed into their raw materials, and those raw materials like lignin raw material or the sugar, the saccharides which the cellulose and hemicellulose are formed of can be converted to these compounds. Of course, because the chemical nature of these three compounds are different, different bioprocessing steps are required. So the way in which cellulose or hemicellulose or lignin is bioprocessed would vary for each other. The same steps, the same mechanism, the same bioprocessing which applies for cellulose cannot be applied to lignin, right? So and this is basically this basically gives the products which can be formed from the biorefinery. This is quite linked to the previous slide in wherein we see the three these are the products based on the individual components. So lignin that can be converted into binders and adhesives and other fuels, while hemicellulose and cellulose, which are made up of pentoses and hexoses, can be converted into again a number of products such as fuels, solvents. There are chemicals, other materials and polymers, lubricants, softeners. So this in totality sums up the complete biorefinery product. You can say which can be produced from lignocellulosic. But in this talk, I'll be specifically talking about lignin, because cellulose and hemicellulose biorefinery is more or less, I won't say that it is well established, but it is quite understood that because these are made up of glucose and pentoses and hexoses, they have been converted. There have been multiple products which have been obtained from them. But lignin as such has still to a very large extent considered as a waste. And much of the lignin that is produced naturally or by industrial processes, it is simply burned down, but it still can be converted into the product. So much of my talk would concentrate on lignin as the material. So now we come to the exact structure of lignin. So as I was telling, it is a cross-linking phenolic polymers, and the monomers, the natural lignin that occurs is actually a polymer of 3 monolignols. And this monolignols are comeryl alcohol, coniferyl alcohol, and synapyl alcohol. So this monolignol structural units these are actually, all these three are the monolignol structural units of hydroxysenamyl alcohols. As you can see, these are methoxylated monolignin monomers, and these monomers then polymerize by various ether and ester linkages to give rise to a highly complex, branched, and varied structure of lignin. So based on these monolignol units, this comeryl alcohol, coniferyl alcohol, and synapyl alcohol, there are 3 lignin building blocks, which can be called as parahydroxy and yeah, okay. So sorry for that disturbance which was there. Anyway, so I was telling about that based on these monolignol units, there are 3 different lignin building blocks, which are hydroxyphenyl, parahydroxyphenyl, guacyl, and syringil, which can be called as the actual building blocks of the lignin compound. So with that, now, what and where exactly would you find lignin? As I was talking that lignocellulose forms, lignin forms a major component of lignin at approximately about 30 to 35%. But it is not just that lignin is present in naturally occurring feedstocks. Actually, the 3 components, the synapyl, coniferyl, and comeryl alcohol, because the composition, the percentage of this different monomeric units in the whole lignin is highly varied. Therefore, in different varieties or in different natural form, you would find that the composition of lignin varies. So in softwood, you would say that in softwood and herbaceous plants, you would have a maximum of coniferyl alcohol. But in hardwood, a major chunk of hardwood lignin is made up of synapyl alcohol. So this is what you can see in the A of the present slide. In B, B gives you the actual value chain for the production of lignin-derived compounds, and this whole value chain consists of three basic processes. One is delignification, followed by depolymerization and upscaling, and exactly in that order. So as you can see, the blue arrows here they show the alkaline delignification processes, and the orange arrows denote the acidic delignification processes. And there are there is dilute acid pretreatment, and I guess this is a hot water pretreatment also. So you can also, the polymeric sulfonated forms of lignin, which you derive from the lignocellulosic biomass, they can be converted into polymeric sulfonated products, such as, and they can be used in low value additives. Now remember, there are low value additives and high value additives. Low value additives are wherein, such as concrete or glue or shock absorbers, fertilizers, photocatalyst supports, emulsions, all these things. But again, these monomeric lignin units can be biochemically converted into high value added products such as phenol, vanillin. These guaiac oil, caffeate, coumarate, ferulate, all these are compounds of higher value. They can also, they can, as a masterpiece of lignin valorization, you can say that these cascade routes produce platform chemicals for commercial plastics such as adipic acid or muconic acid or phthalic acid and they have been reported to be produced at pilot scale. So it's not just it's not that lignin can only, naturally occurring lignin can only be used as a feedstock for animal feeds, I'm sorry, or it can be burned down to produce directly heat energy. But there are a number of compounds which can efficiently be produced. But remember, it's not just that the lignin which is available to us exists in natural form or native biomass, because there is another term which is called technical lignin, and this is the lignin which is produced industrially. So kraft pulping, which is the most common procedure of in paper and pulp industry, the black liquor, the thick tar that comes out contains a majority of lignin. And kraft lignin and soda lignin are the major processes by which technical lignin is produced. And again, as you can see, the aromatic composition of the three monomeric units again differs in that. These processes, whether it is with technical lignin or with natural lignin, they not only promise sustainable green alternatives to fossil routes, but they also enhance the efficiency of existing second generation biorefineries. Now, there is one very important point to note here, that economic considerations reveal a tremendous potential to reduce the price of second generation biofuel and the and also reduce the associated greenhouse gas emission. But this is only possible when there is a better valorization of the lignin fraction of lignocellulosic biomass or technical lignin beyond energy application. So if you just use the lignin for energy purposes, it might not give you 100% output. But if you also incorporate it, if you integrate the whole process for the production of other products also, then you may have then this results in a promising unique win-win situation. For example, in the B diagram, as you can see, with an increase in the lignin conversion to product, there was a significant decrease in the selling price of biodiesel that was produced. And similarly, with a better conversion into product, there was a significant decrease with greenhouse gas emissions also. So this is giving us the unique win-win situation wherein the lignin component is not just used for energy production, but also for production of other chemicals, as you can see here, such as cyclohexane or butadiene or adipic acid or butanediol, which are again very, very important industrial chemicals. Right. So now, before we understand how this lignin can then be converted into various products, the first step that we need to know is depolymerization, how the complex structure of lignin is converted into its monomeric units, and how these monomeric units are then converted into the value-added products. So this gives a thermocatalytic, basically a thermocatalytic process of the lignin depolymerization process. As you can see, there are four steps which are shown here mostly. The first is wherein the non-fractionated lignin, that is lignin directly from lignocellulose via an aqueous phase process through the process of hydrogenolysis can be converted into propyl phenols. Or else, if you fraction out and you purify lignin from the lignocellulosic biomass, you can go through three processes: a dry through a dry process, or a solvent-based conversion, or again through aqueous phase. If you'll see, in contrast to the hydrogenolysis of the native lignin in the non-fractionated biomass, which is shown with this red colored arrow, if you would go with the depolymerization of isolated lignin, so if you isolate lignin from the lignocellulose and then you use this lignin as the starting material for the conversion of products, you can get a more complex mixture and many other aromatic compounds of fuels or biocrude oils like that. So you can say that using fractionated purified lignin component from the biomass is a better option than using the non-fractionated lignin biomass directly, right? So I'm sorry. Right. So when you talk about conversion of lignin to renewable energy, specifically bio oil, there are four major processes by which this can be done, right? So these are the four processes: combustion, gasification, pyrolysis, and hydrothermal liquefaction. Now, out of these four processes, combustion and gasification are exothermic reactions which release energy, which release tremendous amount of heat energy when they're carried out. While pyrolysis and hydrothermal liquefaction, they are endothermic. If we specifically talk about combustion, it is an oxidation process wherein lignin is completely oxidized in presence of air, and whatever amount of energy is released is transferred in the form of heat energy. The major drawback with combustion is it is highly inefficient. So the efficiency is very low, just approximately 10%. Similarly, gasification is another process which involves partial oxidation. And because both of them are exothermic, they are not that effectively used for production of bio oil from lignin. But pyrolysis, if you would see, is a very well-known technique and well-utilized technique for production of bio oil. Now, what happens in actual pyrolysis is the lignin gets decomposed at a higher temperature. The temperature ranges from 300 to 1300 degrees Celsius. And so in absence of air at such a temperature and atmospheric pressure, the lignin gets pyrolyzed to form a large composite, you can say, of carbon, hydrogen, wherein the carbon, hydrogen, and oxygen of lignin decompose into smaller molecules forming gases. Can we have everyone's microphone to mute, please? It is creating a lot of background disturbance.
Yes, sir, we are looking to this disturbance.
Thank you. So what I was saying was, and so by this process of pyrolysis, the large composite of carbon, hydrogen, and oxygen contained within the lignin gets decomposed into smaller molecules forming
Hello.
Am I audible?
Yes, sir, now you're audible.
Again.
Sorry for the disturbance.
No, it's okay. Where was I? So I was saying that this pyrolysis results in the large composite of carbon, hydrogen, and oxygen of lignin getting decomposed into smaller molecules which form gases, condensable vapors, and carbon-rich solid residues. So the solids which result as a result of pyrolysis consists of around carbon and ash. Whatever carbon has been fixed, around 12 to 35%. There's a good amount of gas, around 13 to 35%, which basically consists of methane, hydrogen, and carbon monoxide and dioxide, and a significant amount, that is around 30 to 75%, of aqueous fractions and tar containing liquid. So based on the different temperature and time period of pyrolysis, it has been classified into 4 types, which are slow, fast, flash, and hydropyrolysis. As the name suggests, the slow pyrolysis would have a longer vapor residence time because the heating rate is slow and the temperature is quite less, comparatively 400 degrees Celsius. This results in a comparatively poor quality of bio oil, and it requires high input energy, which is the drawback associated with it. So it was modified to a fast pyrolysis, which is comparatively done at a higher temperature of 500 degrees Celsius. It involves a higher heating rate. The yield and quality of bio oil, which is obtained by fast pyrolysis of lignin, is comparatively good. A better method than this is flash pyrolysis, which then involves a rapid devolatilization of biomass. The heating rate is quite high, approximately Celsius, and the product yield is very, very high, which is around 75%. However, there is one drawback which is associated with flash pyrolysis, which is that the thermal stability is quite poor. And the most, or you can say the choicest method of pyrolysis, which is used for lignin conversion into bio oils, is hydropyrolysis, wherein biomass, or lignin you can say, is heated in presence of high hydrogen pressure. The advantages is the quality of product is extremely high because presence of hydrogen prevents the formation of non-desirable compounds, which can otherwise get accumulated into the final product. So other than pyrolysis, another important process for production of bio oil from lignin is liquefaction. In liquefaction, what we do is we take a batch reactor, and with the desired solvent, and biomass is fed into this batch reactor. The pressure is kept high, approximately 5 to 40 million pascals, and at a temperature of 280 to 370 degrees Celsius is maintained. So the product of this batch reactor is a dark brown tar, which contains of aromatic sugars, oligomers, and hydrocarbons, which can then be purified. Now, the product from this contains around 10 to 20% less oxygen, and the bio oil which is produced has got a high heating value of around 35 to 50 megajoules per kg of dry biomass. So you can see whether it is pyrolysis or liquefaction, the basic process of utilization of this biomass. I have written biomass, but we can say that we are actually talking about lignin here. So in first step, you depolymerize the biomass, which is followed by fragmentation into the various components, and again repolymerization into reactive components, which are of high value, such as oils or other energy products. Right? Now, till now, whatever steps or whatever methods that I have discussed, whether it was pyrolysis or liquefaction or gasification or combustion, all these are industrial methods. Now, still, although very efficient, but because of the high energy demands, because of the high energy requirements, they do not actually satisfy what we call as the green approach for a synthesis of biofuels or chemicals. That is why the concept of biological depolymerization and biological valorization has come up, wherein we actually effectively talk about biologically degrading the lignin structure. As you can see now, this is a very complex figure. I do not want you to concentrate on each and every product. As if you remember, I was talking about the 3 components of lignin, the G lignin, the H and S lignin. So what I basically want to show here is nature has bestowed the microorganisms with enough enzymes and chemicals with which they can actually depolymerize the complex polymer. So whether it is the G-lignin or H-lignin or S-lignin, by various beta oxidation pathways and decarboxylation pathways, it can be converted to, as you can see, protocatechuic acid, and the S-lignin also can be converted into its monomeric compounds. And these compounds can then be utilized into a pathway which can ultimately lead to acetyl-CoA, a succinyl coenzyme A pyruvate, which can be used via TCA cycle into cell growth and product synthesis. So the gist of saying this is, and the summary of this figure says that the lignin monomeric compounds can be effectively used as substrates by a variety of microorganisms, which can, use a number of oxidative pathways to use this monomeric compounds into a significant energy source, right? For this, there are a number of you can say there are a number of enzymes which are available. So if you go on to classify the lignolytic enzymes, either from fungal source or bacterial source, the fungal lignolytic enzymes are of three types, which are peroxidase. They belong to three families. One is peroxidase catalase superfamily. One is copper radical superoxidase (CRO) and GMC superfamily, and the other is multi-copper oxidase superfamily. Of these things, the most commonly studied are the peroxidases from peroxidase catalase superfamily. So there is manganese peroxidase, there is lignin peroxidase. Another very recently identified classes are versatile peroxidase and dye-decolorizing peroxidases. So these dye-decolorizing peroxidases are those which can also help in dye decolorization as well as in lignin depolymerization. And then there is a group which cannot be put into any of these, which is unspecific peroxygenases. The second most commonly studied class of enzymes is the phenol oxidizing lacases. And these are the fungal classes of enzymes which are known for lignolysis. There is another bacterial lignolytic enzymes are another sources. And while most of the bacterial enzymes and fungal enzymes, they overlap. For example, in bacteria also, the dye-decolorizing peroxidases and lignin peroxidases have been reported. But beta etherases are one of the classes which are very unique only to bacteria. So this gives you the overall composition, or you can say the overall overview of the lignolytic enzymes which are present in natural or fungal and bacterial sources. So using those enzymes, now, as we have understood that because bacteria as well as fungi, the microbes, as we can call them, the microbes, they can very effectively utilize lignin. There are again a number of compounds which can be produced by biovalorization of this lignin. So they can be lipids, polyhydroxyalkanoids, cis muconic acids, and aromatic and other compounds. So production of production of lipids is very important because the oleaginous microbes which produce the synthesized lipids can synthesize lipids from the lignin-based aromatics. So lignin, as I've said, is composed of various polymers. These polymers can be used as an energy source, and the oleaginous microbes can use them to synthesize lipids. Similarly, there are various lignin derivatives which can be metabolized into acetyl coenzyme A. And this acetyl coenzyme A is a precursor of polyhydroxyalkanoate synthesis. People who are associated with or who are working with bioplastics, they would know this, that acetyl coenzyme A is a very important and a target molecule even in genetic engineering for PHA synthesis. So here is wherein you can incorporate these two things and then can utilize this lignin for production of something called as bioplastics by PHA. Cis muconic acid has come up as an excellent byproduct from lignin biovalorization, because it has recently been identified to be an intermediate of adipic acid production, and it is also being used for as a bulk feedstock for production of various other biofibers and plastics. And probably the most important or the most significant and the largest class of value-added products which are produced from lignin are the aromatic and the biopolymers, because this lignin monomeric units, they appear as an ideal feedstock for natural aromatics. So we would specifically concentrate on the aromatics that are derived from lignin, although there are a variety of biopolymers which can be produced. The most only till date, only 3 of them are industrially produced from lignin, which are vanillin, para-hydroxybenzoic acid, and pyrogallol.
You sir, can you just we have a little bit of short time, so can you make it a little faster, like maybe?
Yeah, sure. I'm just finishing.
47. Yeah.
I was just saying that all vanillin, PHB, and pyrogallol, these are the compounds which have been produced, and they have attracted most attention from for the production of aromatics. Vanillin, as you know, is an organoleptic ingredient of the vanilla, and it is widely used in food, cosmetics, pharmaceutical, and other industries. But because there is a limited supply and there's a great demand, most of this demand is compensated by the chemical conversion of petrochemicals, which is again non-sustainable. So lignin conversion to vanillin could be a very important pathway that can be further explored. Again, for polyhydroxybenzoate, which are monohydroxybenzoic acids, which have good antimicrobial and antioxidant properties, and they have low toxicity, and they are used as preservatives in food, flavors, cosmetics. So these are the other avenues which can be explored. I'm audible, I suppose, and my slides are visible?
Yes, sir.
Yeah.
You are audible and your slides are also visible.
Yeah. So lignin-derived polymeric materials can be classified into these 4 classes. This is available in much of the literature. I'll not go into details of this. What is important is, this shows the polymerization strategies. Then again, this can be read in literature. I would very fast. Okay, so this is where I was talking about what is the biovalorization. So, and this gives you the strategy. So we have lignin, we can depolymerize lignin. Once we have depolymerized it, the monomers can be used by the bacteria and they can be assimilated, and you can have value-added products. So this is the summary, which is giving you how lignin can be used for the value-added products. There is one significant application other than energy and other than production of chemicals also, and that is in drug delivery. As you can see, I have shown here the 2 mechanisms. So if in diagram A, as you can see, the silver nanoparticle has been bacteria an antibacterial agent and remains into the environment. But silver ion infused this environmentally benign nanoparticle, which have a cationic polyelectrolyte coating over them. They can easily bind to the cell walls. And in contrast to silver nanoparticles, these environmentally benign nanoparticles are very soon depleted of silver ions during their application, and therefore their post-utilization activity is minimized, and the inactive particle which is produced, which is left out, is biodegradable. So this was it, and now. See, the most important aspect is the systems biology approach for lignin valorization. And as I've said, this actually uses the omics technology for the production of, and it guides the synthetic biology towards lignin valorization. So there is genome analysis, which would relieve the potential mechanism in lignin degradation. There is transcriptomics, which can provide a blueprint of the genes, pathways, and microorganisms for lignin degradation. Similarly, there is proteomic analysis, which would give the protein abundance and which is important for the protein activity and functionality. or the metabolomic study, which would help in analyzing the metabolites and the unique chemical fingerprinting prints and system level mechanisms for lignin degradation. So the concept is, overall, this omics based study would be crucial to achieve the exhaustive understanding of molecular and system level lignin degradation in microorganisms. So all that said and done, there has been a very common saying which goes like this, that lignin can produce everything other than money. And the reason is, for a very long time, when lignin was converted into various products, the process was not economically efficient. So a techno-economic analysis is very much required to understand how you can achieve the goal cost, or how you can make the process economically efficient. So there are various process parameters such as temperature, pressure, and all these. And these process parameters can be identified from the already published literature or the empirical bench and pilot study experiments that has been done. This gives you a conceptual process design, which ultimately leads to a material and energy balance, which can be based on the capital and the material costs and the operation cost. You can estimate the capital and project, the total cost of the project. You can also you must also have into account the environmental and the sustainability analysis based on the raw materials and yields and the emissions that are there. And this ultimately leads to the economic analysis and logical economic analysis. And if all these steps are taken out, are carried out in a logical manner, you can have your exact goal cost. And this actually helps in estimating the total cost, and whether the process that has been designed based on these parameters would be techno-economically feasible or not, whether it will be sustainable or not. Because understand the bottom line of everything that has been said and done till now. If the process is not economically sustainable, if it is not economically profitable, it will not exist in market. Whatever product that you are going to do will not enter into the market. So with this, I come to the end of my talk. These are the major prospective areas that still need to be explored to have an efficient biorefinery process. We still need to identify the authentic structure of lignin that occurs after various pre-treatment methods. We also need to have develop the new fractionation methods that can deliver high purity sugars and native lignin streams at the same time. And we also need to decipher the structure activity relationship between lignin and its valorization products. So all this, if we can achieve, we can dream of a lignin biorefinery, which is actually giving us value added or commodity products from the waste material. With that, I would like to thank all of you for attending me, for listening to me so patiently. I would leave you all with this thought. If you want to increase your success rate, double your failure rate. Now, it is up to you to ponder over this thought. I'll give a rest to my voice. Thank you. Thank you very much for listening to me. Thank you.
Thank you so much, sir. That was such an erudite session for all of us. Now, as we are receiving so many questions from our participants,
I would like to answer a few of them.
Unfortunately, we cannot take all of that because of the time constraint, but I'll take one question from our participants. There's one question that, from Dr. Prasanta Dhea. Hydrogenolysis of complex cellulose leads to production of very limited products. But if we increase the pressure, can the temperature be lowered to get the maximum number of byproducts?
You see, in all thermocatalytic processes, in all such industrial processes, whenever as Sir was talking about hydrogenolysis, you can go for, there has to be a balance which has to be kept between pressure and temperature. So even if you are lowering, can you please repeat the question? You are lowering the pressure or increasing the pressure to reduce the temperature.
By increasing the pressure, can the temperature be lowered to get the maximum number of byproducts?
But only to a limited extent, It cannot be that you can increase the pressure so much that, it would again, there has to be a balance which has to be kept between the pressure that you are applying and the temperature, because it is a play of both of these processes that ultimately leads to depolymerization. So there has to be a balance. You cannot go beyond a particular limit of lowering the pressure.
Okay, thank you so much, sir. Thank you for your informative lecture.
Thank you very much once again to all the organizers for giving me this opportunity. Thank you, Kanupri, also for organizing or managing this session for me. Thank you.
Thank
Thank
you
you
so
very
much.
much.
Thank you so much.
Thank you, Dr. Piyush, for such a nice presentation. Thank you very
Thank
much.
you. Thank you. Thank you. Thank you, Soumya. Thank you so much.
So now we have our next speaker today, that's Dr. Rui Miao. She's a postdoc in CEA, French Atom Energy and Alternative Energy Commission. Dr. Miao completed master degree and PhD both from Uppsala University in Sweden under the supervision of Professor Peter Lindblad. Dr. Miao's research is about using the photosynthetic microorganism cyanobacteria to produce valuable chemicals, including biofuels. She has first authored scientific publications in several high-impact journals, including Energy and Environmental Science, Metabolic Engineering, Biotechnology for Biofuels, and Current Opinion on Biochemistry. We welcome you, ma'am.
Hello.
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 Over to you, ma'am. We can hear you.
Okay. I will share my screen now. A moment. Share screen. Okay. Yeah. Hi, everybody. My name is Rui Miao. First of all, I would like to thank the organizing committee for the invitation for me to give the talk here today. One moment, sorry. So today I will talk about the biofuel production using cyanobacteria as a chassis. First of all, I would like to introduce a little bit background about myself. I got my PhD degree in 2018 in Uppsala University in Sweden. And then I moved to France to do my first postdoc there in CA, the Atom Energy and Alternative Energy Commission. And it's a very interesting place in France because we are in the research center together with the nuclear researchers. So that's why I cannot give you a full view picture of the research center because we cannot see it from Google Map even. Yep, so now I will start my talk today. So why do we need biofuels? I guess all of us know there is a huge increase of energy demand nowadays because of the increased amount of electricity usage and fuel needed for the needs for car driving and other transportations. And also there is an environmental demand to use biofuels because when we burn oil or other fossil fuels, not only CO2 is produced, but also many other harmful particles are produced at the same time. And these particles are extremely harmful to human health. And there was a statistic study showed that nearly 4.2 million deaths was caused every year because of the outdoor air pollution. And another very important reason to develop biofuel production is that we really want to decentralize the energy resource nowadays on the Earth. And also we want to decrease the geographical restriction of the resources. As we know, nowadays almost 1/3 of the international conflicts are because of the resources like oil and water. So if we want to solve that problem, we need to put much more attention on biofuel development. So for our research, we use photosynthetic organisms to produce biofuels. Why? Because there's a plenty of solar energy reach the surface of our Earth every day. For example, the energy reach the surface of Earth per hour is more than enough for the human energy consumption for entire year. But the solar energy we can use nowadays is very limited. So we are wasting huge amount of solar energy. On the bottom, I showed the energy supply and consumption in Sweden in 2017. Here we can see in Sweden, there's quite a large percentage of energy is produced from biofuels. And also they use lots of nuclear fuel. But this is not the case for most of the countries in the world. And they have much less percentage for biofuels production, energy supply. So we have a long way to go here. And for the photosynthetic organisms, they are very economical to use, because if you provide them CO2, water, and sunlight, then they can become a small photosynthetic factory to produce whatever fuel we want. And the organism I was using in my PhD study is cyanobacteria. They are a group of gram-negative probiotics that live in a variety of environments. You can find them almost everywhere on the Earth, from fresh water to seawater, even in rocks and deserts. And they are the very first cause of the oxygen on the Earth. That was almost 2.5 billion years ago. And there are many different kinds of cyanobacteria strains. Here I showed three pictures: the filamentous strain and unicellular strains, and there are also some colonial strains, which I don't show the picture here. And the very important reason we use cyanobacteria is that they have 3 times higher solar energy capturing efficiency if we compare them to higher plants. And this is because they have a very efficient CO2 and bicarbonate uptake and concentrating system in the cell. And this system can accelerate the photosynthesis process. And also another reason we use cyanobacteria is they have relatively simple and transformable genome. So up to date, there are many studies going on to develop genetic engineered tools to manipulate the genome of different cyanobacteria strains. And also these microorganisms are relatively easy to grow. They can tolerate very high salinity and wide range of pH and temperature, and also different light intensity. But we need to be very careful when we choose the chassis strain, because some of cyanobacteria are toxic. They produce toxin to the water, and which can be harmful for human and animals. Specifically, the cyanobacteria strain I was using is a unicellular strain called Syncocystis, PCC 6803. It is a model freshwater cyanobacterial strain and was fully sequenced in 1996. And it was the first photosynthetic microorganisms that was fully sequenced. It has a simple genome, and also it has the ability to uptake foreign DNA into the cell via different methods like native transformation or conjugation. As I said before, there's been intensive research going on to develop the genetic tools for metabolic engineering. Here I showed the transmission electron microscopy picture of the Syncocystis cell. Here we can see the wave inside the cell is the thylakoid membrane. It represents the cartoon here, and this is where the photosynthesis happened. So for cyanobacteria, they have 2 photosystems, photosystem 2 and system 1. When you shine light on the cells, they produce oxygen, ATP and ADPH. And ATP and ADPH are the driving force of the Calvin-Benson cycle. And then the sugar that produced from the Calvin-Benson cycle will be the precursors for all the other metabolic pathways inside the cell. So the biofuel we wanted to produce inside synecocystis is butanol. Why do we want to produce butanol? Because butanol can be a promising gasoline substitute because they have relatively high energy content, which was similar to that of gasoline. And also they don't absorb water from air, which makes them a perfect drop-in fuel. And also they are very safe to use. And another reason, especially for the countries like in Sweden, you always have difficulty to start your engine during winter. So if you blend butanol in gasoline, or you use pure butanol as fuel, then it's much easier for your life. Another important reason that we produce butanol is that the use of butanol does not require any modification on gasoline engine exhaust system. So this is very important for the car industry. So nowadays, the butanol mainly synthesized through chemical process, and it's very expensive. So it's not feasible to use butanol as a fuel for our cars or trains. So we need to figure out a cheaper way to produce butanol. So here I show the simple figure that represents our workflow. So when we decide which chemical we want to produce in cyanobacteria, we go in for a bioinformatics study to find out the metabolic pathway we want to manipulate with. And then we design our metabolic pathways, the artificial metabolic pathways, and then we construct the operon, or the entire pathway in E. coli first, and then we extract the DNA from E. coli and put the DNA together with synecocystis, and then synecocystis can directly take up the foreign DNA into itself. And then after selection and cultivation, we have the culture which can produce the target chemical, and then we detect the product. If we are not satisfied with the results, we will go back one more round to optimize the entire pathway and all the cultivation condition to improve the productivity in the end. So from now, I will go to some details about isobutanol and 1-butanol production in synecocystis. As we know, synecocystis does not produce neither of these two products. But interestingly, the cell has intermediates called 2-keto-isovalerate, which which belongs to the amino acid synthesis pathway. Actually, this is a precursor for isobutanol production. But synecocystis doesn't have the KIFD enzyme here to catalyze 2-keto-isovalerate to isobutyraldehyde. But synecocystis does have the ADH, the alkyl dehydrogenases, naturally. But it's not for isobutanol production, it's for ethanol production. So in the beginning, we were not sure if the native ADH in synecocystis can use isobutyraldehyde as the substrate. So we needed to try. So our initial plan was to introduce a KIFD from Lactococcus lactis, and also one of the alkyl dehydrogenases from different organisms into the cell. So 2 native ones and 2 highly active ones from E. coli. Therefore, we made the construct, 4 constructs. For each construct, there is KIFD and one of the ADH. And these constructs was put into the genome of synecocystis. And then we examined the transcription and translation level of the enzymes. Apparently, the translation level was not really high, which resulted in a very low isobutanol production in the end. Then we have to improve. And so on the next stage, we change the transcription. We change the promoter in the operon to a stronger one. And also, we try to put the operon onto self-replicating vector. That means we don't change the genome of the cell, but we put an extra DNA piece, circular piece into the cell. Because for some living conditions, the cells can have more copy number for the self-replicating vector than the genome. So we expect it to have higher expression level for the operon on the self-replicating vector. And we got positive results. We saw more protein, and also we saw much more isobutanol titer, but we were not satisfied either with this number. So we need to improve more. To improve more, we thought because syncosystis has native alkyl dehydrogenases, then we made another construct with only KIF-D enzyme on it. And surprisingly, this enzyme, this construct gave higher isobutanol production per OD compared to the other 4 with extra copy of alkyl dehydrogenases. So we took the string with only KIF-D for the further study. Because more, we realized from the previous results, we realized that more KIF-D, more isobutanol production. That means KIF-D can be a potential bottleneck in the metabolic pathway. So how to overcome the bottleneck? Then, if we look into the substrates for KIF-D enzyme, we realize that there were actually KIF-D does not only use 2-ketylisylvalerate as substrates, but also ketylysylcopperate. And that's why we always see a byproduct, a 3-methyl-1-butanol, together with our main target product, isobutanol. So that means KIF-D is using both substrates in syncosystis. And if we look at the 2 substrates, we know that the KIC, the substrate for 3-methyl-1-butanol, is much bigger than the KV, the substrate for isobutanol production. Then that means we have to make the enzyme take more of the small substrates instead of the big one, in order to make more isobutanol here. And then we decided to make fractional design to engineer the KIF-D from Lactococcus lactis. But unfortunately, we didn't have the crystal structure for this specific enzyme, but we did find the crystal structure for another substrate, substrain of Lactococcus lactis. This enzyme shares 88% amino acid sequence as the enzyme we are using. So that gave us very good information to look into the active site and the substrate binding pockets of the enzyme. If we want the enzyme to take more small substrate, that means we need to shrink the substrate binding pockets. So in the end, we had 12 different KIF-D design. For each variant, we changed 1 amino acid. We changed 1 amino acid involved in the substrate binding pockets. And then we look at the translation level of the different KIF-D variants. Apparently, it differed quite a lot. For some mutants, we got a lot of protein, but for some mutants, we didn't see at all the protein. Among all of these mutants, we got isobutanol production from 6 of them. And so it's very it surprised us quite a lot that the mutant ST and VI had huge increase for both isobutanol titer and the byproduct 3-methyl-1-butanol titer. And also the double mutant SV, which is a combination of the mutant VI and ST, showed the highest isobutanol to 3-methyl-1-butanol ratio. But the total titer was kind of low. But anyway, we took these 3 strains for further study. And then we monitored the isobutanol production along 8 days cultivation. The same result as before. We had the highest isobutanol production from mutant VI and ST, and the double mutant showed very low isobutanol production and also titer. But as I said before, the SV showed the highest isobutanol to 3-methyl-1-butanol ratio. We also performed in vitro activity assay for the engineered enzyme. And for all the 3 engineered KIFDs, 2 single mutants and the double mutant, all of them showed preferential shift towards isobutanol production. But unfortunately, the double mutant with the highest preferential shift showed the lowest expression level, which led to the lowest in total production. So we took the best isobutanol producing strain for long-term cultivation under light, and the bicarbonate is the carbon source for the cells. And so after doing this, we achieved the maximum cumulative isobutanol titer of 911 milligram per liter at day 46. And the fastest producing period is here around day 5, and which is for nearly 45 milligram per liter per day productivity. And so up to date, this number, 911 milligram per liter, is the highest photoautotrophic isobutanol production in all cyanobacterial strains. So here is a small summary of the isobutanol production in Synecocystis. So we try to optimize the production from transcription level, translation level, protein level, and also cultivation condition level. In the end, we achieved the 84-fold increase of isobutanol production after doing all of this. Another part of my thesis was 1-butanol production in Synecocystis. We went through the similar strategy as the isobutanol production, but because 1-butanol synthesis pathway is much more complex than the isobutanol pathway, because we have 6 steps from acetyl-CoA to the final product 1-butanol. So we have to introduce 6 genes into the cell. That's quite challenging for cyanobacteria. We divided these 6 genes into 3 different operons and put them into 3 different loci on the genome of Synecocystis. And then we screened the different strains with different enzymes, and we selected optimized one, show the highest 1-butanol production. And also we did some protein engineer for some of the enzymes, and we again we selected the most active one for further study. And then after choosing the enzymes for the pathway, we played a little bit around the expression level of the enzymes, because we want to increase the expression level as much as possible. So we tried different promoters and also 5' UTR 5' UTR modifications, and to increase the translation level. And then in the end, we got a strain that had the highest expression level for all the 6 genes. And also we got the highest 1-butanol production for that strain. So we took this strain for a further step. So after we optimized the metabolic pathway, we wanted to increase the precursor amount in the cell. That is the acetyl-CoA. As we know, the acetyl-CoA pool in cyanobacteria is very limited. So we wanted to introduce an enzyme here, PK, to drag more carbon source from CBB cycle directly to acetyl-P. And then we overexpressed the gene converting acetyl-P to acetyl-CoA. So in the end, we expected to have more acetyl-CoA in the cell. So we tried 9 different PK genes from different organisms. And after monitoring the expression level of the enzyme and also 1-butanol production, we chose the best candidates. So after having the optimized strain, we also did the long-term cultivation for this strain. And also we tried to provide a carbon source at different time points. So for one group, we provide bicarbonate every 48 hours, and the other group, we provide it every 24 hours. And then by comparing those 2 groups, we got the maximum cumulative 1-butanol titer of 4.8 gram per liter from the 24-hour group, which is the highest 1-butanol production in cyanobacteria up to date. So if we compare the isobutanol and 1-butanol production from cyanobacteria to the ones produced in E. coli and yeast, which are the traditional microorganisms for producing biofuels, we can see the titer for cyanobacteria is closer to that of yeast, but is very far away from the production from E. coli. Mainly because E. coli is a very well-studied organisms, and there are plenty of genetic engineering tools to modify the cell as much as we want. But for cyanobacteria, it's very limited. So we have to develop more tools to be able to boost the expression level of all the metabolic pathways. And also, we need to find more robust cyanobacterial strains, which can grow much faster, and also which can tolerate more tricky environments. And then we will be able to grow the cells with the industry exhaust gas and also wastewater. But in general, I think cyanobacteria producing biofuels is compatible to the ones produced from other organisms, and also it's feasible in the near future. So in the end, I would like to introduce you my PhD supervisor, Professor Peter Lindblad, and also 2 of my co-supervisors. And all of these are the research funding for my PhD study. Thank you very much. Thank you for your attention.
Thank you so much, ma'am. Thank you for your time, and thank you for these valuable words. We have one question from the participants.
Yeah.
This question is from Yogesh Nagwekar. He said, as it shares some structural similarity to pyruvate decarboxylase, so can it be used as a catalyst?
Yes. You mean in vitro catalyst? I think it's possible.
Okay.
Yeah.
Okay. Thank you so much, ma'am.
Thank you.
Thank you for your time. Now we welcome our last speaker of the day, that is Dr. M.V. Reddy, who could not deliver his talk yesterday due to the technical issues. So now I welcome Dr. M.V. Reddy, who is currently working as Alexander von Humboldt postdoctoral fellow at Westphalisch-Wilhelms University, Münster, Germany. Previously, he worked as JSPS postdoctoral fellow at Muron Institute of Technology, Japan. He completed his doctoral thesis in 2014 at CSIR IICT, India. His research focuses for the generation of biofuels and bioplastics using advanced microbial fermentation technologies. His research was disseminated in 38 SCI journal articles with H index 20. Now I welcome you, sir.
Yeah, hello everyone.
Hello, sir. Over to you.
Yeah, can you see my screen?
Not yet, sir.
Yeah, now I think now.
Uh, yes, you have to double click. Yes, sir. Now it is visible.
Yeah, okay. Yeah, good afternoon, everyone. I would like to thanks the organizers of Waste to Bioenergy conference, and also my special thanks to Dr. Soumya Pandit for giving me this opportunity. Myself, I am Motakatla Venkateswar Reddy. I am originally from Hyderabad. I did my PhD in 2014 in CSIR Indian Institute of Chemical Technology in India, and presently I'm working as a postdoctoral fellow in Germany. So where I am working now, I'm working in the university called Westphalisch Wilhelm Universität, Münster. The university was located in the city named as Münster. You can see here the map. So this university is Germany's fifth largest university with more than 43,000 students, and it has over 120 fields of study in 15 departments. So here you can see the administrative building of the university. So actually the university was located in all over the city. So it means in the city, in everywhere, it has its own building, like economic faculty in one place, science and biotechnology faculty in another place. So it's not like in India or maybe in Japan, we have one big campus. So it has spread all over the city, all the departments. So you can see my title of presentation is polyhydroxy alkanoates. So I hope many of you know what are polyhydroxy alkanoates. And in this presentation, I will mainly cover the why we are producing polyhydroxy alkanoates, what are the advantages of producing these polyhydroxy alkanoates. If by chance, if some of you don't know what are polyhydroxy alkanoates, I will give you some brief introduction. So these polyhydroxy alkanoates are one of the raw materials used for the production of bioplastics. So many of you know why we are producing bioplastics. Even though the normal plastics like polyethylene, polyvinyl chloride are available in the nature, and again why we are producing bioplastics. So in this slide, I would like to give some introduction about what are the normal plastics and what are the problems caused by these normal plastics. So the normal plastics are high molecular mass organic polymers. They are synthetic, derived from petrochemicals, and are non-degradable. But some plastics may be degradable, but it takes hundreds of years for their complete degradation. And the only main method many researchers using for the degradation of normal plastics is open air burning. But the main disadvantage is open air burning of plastics releases some toxic gases, and these toxic gases show some toxic effects on human and environment. And also these gases are carcinogenic and they will cause the cancer, and they will changes the gene sequences in aquatic animals and also in human beings. So it causes mutations in genes. Also, these toxic gases will affect on hormonal system, reproductive system, blood cells, and nervous system. And normally for the production of these plastics, many additives are used, but these additives are they will cause many allergies to human beings. And here in this slide, we can see what are the problems with the plastics. So here you can see the seal, how the seal neck was surrounded with non-degradable nylon plastic. So you can imagine what happens here. This nylon was not degraded, so it will cause the death of seal after some years. Also here you can see the how tortoise was affecting with the plastic. And here you can see the bird in the inside stomach. We can see some plastic caps and plastic bottles, small bottles. When we throw some plastic bottles or caps in the seaside or in the beach, then these plastic caps are floating on the water, and birds think it is some food material, and they will eat them. And after going inside the stomach of bird, these plastics are not degradable, and it will cause some health effects and ultimately the death of animals. Also in these pictures, we can see how plastics are accumulating around the sea and beaches in many places. So now you can understand what are the problems with these normal plastics due to their non-degradable nature. That's why many researchers are focusing on the production of bioplastics. Why they are producing bioplastics? Because compared with synthetic plastics, these bioplastics are biodegradable. In this picture, you can see how the bottle made with bioplastic can be degradable. Here, the first one is at 0 day, and then at the end, after 6 weeks, almost 30 to 40% of bottle can be degradable. And these bioplastics are made from biological sources such as plants and bacteria, and they are degradable and are environmental friendly. If you look at synthetic plastics, open air burning will release carbon dioxide, and it is not at all environmental friendly gas. And also these bioplastics reduce synthetic plastic waste, and it will reduce the emissions of greenhouse gas, and they protect environment, aquatic and terrestrial animals. They are degraded by the action of fungi, bacteria, and enzymes. So in this picture, we can see the some of the products, how they are produced by using bioplastics materials. Here you can see actually this bag is from German company. You can see Bioprodukte, and also some cap, some covers and glasses, spoons, and some of the material which are made with biodegradable plastics. Actually, researchers are working for the production of many varieties of bioplastics, but I'm not going to in detail because my area of research interest is production of polyhydroxyalkanoates. So these are very interesting materials because these PHA have some of the structure similarities, and also some of the properties have similarity with the normal existing uh, uh, plastics. So here you can see the different types of bioplastics like polyhydroxyalkanoates, polyhydroxybutyrate, polyhydroxyvalerate, and polylactic acid. So from last 6 to 7 years, I am mainly working for the production of polyhydroxyalkanoates, polyhydroxybutyrate, and polyhydroxyvalerate. So initially, researchers are used to some plants like corn for the production of these bioplastics, but due to now due to lack of land and due to usage of this land into edible plants growth, we cannot use these plants for the production of plastics. That's why researchers move to bacteria. When we use bacteria for production of these bioplastics, we no need to depend on land. So we can grow the bacteria in the fermenter inside the laboratory. So here I will explain the clear methodology, what we are doing for the production of PHA. Actually, initially we focused for the production of PHA using some synthetic substrates like glucose, acetic acid, and some other fatty acids. But after that, we realized that utilization of these synthetic acids and commercial substrates are enhancing the bioplastics production cost, because we need to pay some money to buy the glucose or some other acids. That's why we moved to utilization of some waste materials for the production of bioplastics. So in this picture, we can see mainly we collect the waste material from municipal and agricultural waste. So after collecting them, we will store these waste materials in cold room, because the same waste material we can use for 1 or 2 years. And then after collecting from the source, we will move to storeroom, and then in the from cold room we will bring to laboratory, and then we will do anaerobic digestion of this waste material using the anaerobic mixed culture, because this anaerobic mixed culture have the special property of doing anaerobic digestion, and through this anaerobic digestion process bacteria produce short chain fatty acids, like from carbon 2 to carbon 5. I think many of you know what are short chain fatty acids. Those are like with the fatty acids which have straight chain, and from carbon 2 to carbon 5. For example, acetic acid, propionic acid, butyric acid, and valeric acid. So many researchers are working for the production of these short chain fatty acids, but the main disadvantage is these are easily soluble in media or in the wastewater. So after production of these short chain fatty acids, it is very difficult to extract them from the media. Of course, there are some methodologies available for the isolation of these short chain fatty acids, but it's a really energy expensive method. That's why in our experiment, we are converting these short chain fatty acids into polyhydroxyalkanoates. Polyhydroxyalkanoates are not like short chain fatty acids. These are solid materials, and these are not at all soluble in the liquid media. These PHA are accumulated inside the bacterial cells as a solid material. So it is easy to separate them from the media. And also these polyhydroxyalkanoates have many applications, like we can use them in the biomedical applications. The main application is packaging, like we can prepare some plastic cups, plastic covers, and many things we can prepare by using this PHA. And also these are useful for nano composites preparation, electron beam cross linking methods, and also plasma surface treatment method. By speaking frankly, even I don't know how we can use this PHA in electron beam cross-linking and also for plasma surface treatment methods. But my main target is to produce some packaging materials by using this polyhydroxyalkanoate. Also, we can use this PHA in biomedical application. When we use a pure substrates like acetate or glucose, then we can get some pure PHA, and this pure PHA we can use for the production of in the biomedical application. But in our research, we are using waste materials, and we are producing PHA from waste materials, and the produced PHA has some impurities, because wastewater contains thousands or hundreds of waste materials. So these waste materials will also come into the structure of PHA. So we cannot use the PHA produced from waste materials in biomedical applications. But if you want to use PHA in biomedical applications, the main thing is we need to use some single bacterial culture and also single substrates, single and pure substrates like acetate or glucose. Also in this slide, we can see the bioplastics production from wastewater. Actually, this work I done when I was in Japan. In there, we collected wastewater from different sources. Here you can see the Muroran. Actually, it is a one city name. So we collected wastewater from Mudoran wastewater treatment plant, and also the second one is Dhea. It's also our neighboring city. We also collected the wastewater from Dhea wastewater treatment plant, and also Nobori Betsu wastewater treatment plant. Actually, these three cities are located in northern Japan, the state named as Hokkaido. So in this slide, you can see the wastewater we collected from Nobori Betsu wastewater treatment plant. So after collection, we just pour this wastewater in beaker to see how it's looking. Here you can see how this wastewater before treatment, and then after that we added this wastewater as a substrate into the bacteria, and we added in aerobic fermenter, and this bacteria also aerobic bacteria. After fermentation process, usually it takes 2 to 3 days. Then we produce the PHA. Here, this PHA was extracted and purified using methanol precipitation method and also chloroform extraction method. If you want more details about how we extract these PHA, you can contact me in email because it's a really long protocol. So in email, I can suggest you some good publications. So about the protocols, how we can purify and how we can extract the PHA polymers from bacteria. And you can see here the wastewater after treatment. And also this work also done in Japan. So actually in this slide, we collected almost 25 types of wastewater from different sources, mainly from Japan and Korea. Here you can see from Korea and Japan. At this time, we have a collaboration project from Korea also. From Korean researchers, they collected some wastewater in Korea, and also from my side in Japan, we collected wastewaters in Japan. So here I can give you some examples of wastewaters, what we use. For example, here you can see LF. LF means livestock farm. Also, you can see PFPF. Here, yeah, PFPF means preserved food production factory. Also, NCW means Nobori Betsu Cheeseway Factory. So here you see the abbreviations, and also in the down section you can see the full forms of the wastewater industries we collected wastewater. Among 25 wastewaters, bacteria with some of the wastewaters showed good production. For example, here you can see the Nobori Betsu Cheeseway factory, bacteria produced 71 milligram per liter of PHA. Then actually this cheeseway wastewater has some very high COD. Then in next experiment, we just diluted the wastewater to 15 times, 15 times dilution, and then we load this wastewater into bacteria. Bacteria showed good production with diluted cheeseway, and the production was almost double. You can see without dilution, it is a 71 milligram, and after dilution, it is 151 milligram per liter. And also bacteria showed good production with Dhea sewage treatment plant. It is about 145 milligram per liter. Unfortunately, bacteria was unable to show production with some of the wastewaters. Like here, you can see zero. So zero means no PHA production. It might be due to the composition of wastewater, because each wastewater has a different composition, and some wastewaters may contain some toxic materials, and this toxic material will show some negative effect on bacterial growth. Also, in the right side here, you can see the transmission electron microscopic image of the PHA granules accumulated inside the bacterial cell. After production of this PHA, I already told we will extract this PHA from the bacteria using sonicator and rotavapor, and also we used some solvents like sodium hypochlorite and chloroform. After extraction, again we will purify using cold methanol. This purification was done through precipitation methodology. So after that, this purified PHA was used for the structure determination, because we need to know what is the polymer composition, how it looks, and what is its structure. Also, we will do some properties like melting temperature and decomposition temperature. So I will explain those in my next slide. So in this slide, you can see the structure of PHA produced from various wastewaters. So here you can see P3HP standard. That means this P3HP was actually we buy from Sigma Aldrich company, and then we done proton NMR structure analysis for this standard, and then we also did NMR analysis for the samples collected from different sources like wastewater from Dhea city, Cheeseway wastewater, and also wastewater from Muroran city. If you see here, the NMR spectrum was almost similar with the standard PHP. That means the produced polymer is has the same structure with the standard P3HP, and also here you can see the carbon 13 NMR spectrum. In proton NMR spectrum, we know the proton arrangement of in polymer. In carbon 13 NMR, we know how carbon was arranged in the polymer structure. So the same was done with C13 NMR also. First we analyzed the structure of P3HP, and then the PHA extracted from different wastewaters. But in PHA extracted from different wastewaters, we can see some extra peaks. These peaks were due to the additives and some pollutants present in wastewater. So these toxic and pollutants also coming into the PHA structure, and it shows some extra peaks in the structure of the polymer. And also you can see the disturbances in all three wastewaters. And here this is a wastewater collected from other source like Muroran wastewater treatment plant. Actually, this wastewater looks very dark when we collect it, and then after that we added aerobic bacteria and this wastewater in aerobic fermenter, and we produced the polymer. And then here you can see the treated wastewater, and this wastewater we can use for purposes like gardening. We cannot use this wastewater for drinking purpose, but we can use it for gardening purpose. Also here in our next experiment, actually we used some toxic compounds like 4-butyl phenol, 4 secondary butyl phenol, 4 tertiary butyl phenol, 4 tertiary octyl phenol, and 4 nonyl phenol. Actually these are kind of alkyl phenols. These alkyl phenols are present in many industrial wastewaters, and they will cause some adverse effect on environment. So our aim is to degrade these alkyl phenols and also simultaneously convert these alkyl phenols into the useful product like PHP. So in this graph, you can see the PHP production in percent dry cell weight by using various toxic compounds. Here we used the bacteria Cupriavides species CY1. Bacteria with phenol showed 48% dry cell weight of PHA, and also with the naphthalene, it showed 42% dry cell weight of PHA. But bacteria was unable to show PHA with some of the toxic compounds like 4-butyl phenol and 4-tertiary butyl phenol, and also like phenanthrene. And at the same time, we captured the transmission electron microscopic image of the thin sections of the bacteria. Here you can see the A is when bacteria grown with phenol, and from B2 is when bacteria grown with naphthalene. So here you can see the PHA granule accumulated inside the bacterial cells. And also we analyzed the structure of the polymer produced from phenol. And at the same time, we done proton NMR analysis using the standard PHP. So in the right side is a standard P3HP, and left side is a P3HP extracted from the bacteria when bacteria was incubated with phenol. So the both spectra were looking similar. So the produced polymer was P3HP. Also, we done the carbon 13 NMR to know the carbon arrangement in both standard and sample P3HP. Yeah, in my previous slides, I told along with the structure similarity, we also evaluated some physical and thermal properties like melting temperature and decomposition temperature. So here we evaluated the melting temperature using differential scanning calorimetry. So this instrument will give you the melting temperature of each polymer. So here in the right side top, you can see the melting temperature of P3HP, the standard P3HP. It has a 178 degrees centigrade, but the P3HP extracted from sample has a melting temperature of 160 degrees centigrade. And if you clearly observe the difference between standard and sample, in standard you see only one big large peak, but in sample you can see many peaks. It means along with the PHP, there are some other components present in the polymer structure, because this standard PHP is a highly pure PHP, but in our sample is it's not that much purity, even though we used methanol precipitation method for purification. So in our future experiments, we need to think about how we can get highly pure PHP. Also in the down, you can see the decomposition temperature of PHP extracted from the sample and standard. Here the dotted lines are the decomposition temperature of standard PHP. Here you can see at 300 degrees centigrade, the standard was completely degraded. But if you see the sample, it was not degraded even at 600 degrees centigrade. So yeah, I already told the sample has some impurities. That's why it is not completely degraded. So this is a work I done when I stayed in Korea for my first postdoc. There actually we used some toxic compounds. Actually these toxic compounds are, here you can see the monoaromatic compounds, and left side you can see the structure of lignin. So lignin has very highly complex structure. You can see here the many aromatic compounds are linked and it form a rigid structure. After degradation or hydrolysis, or after using some enzymes like demethylation enzymes, we can degrade the lignin, and this degradation produces some aromatic compounds. Actually, I am not expert in degradation of lignin, but I know the methodologies of how we can degrade lignin by using some special enzymes like demethylases. What we done, we purchased the monoaromatic compounds from company, and the commercially available monoaromatic compounds were used in our studies. So our aim is to use lignin degraded monoaromatic compounds and produce energy by using the bacteria. So for this, initially we used the bacteria named Comomonas testosterone, and also different monoaromatic compounds were fed to this bacteria to check the growth, whether this bacteria can degrade these toxic compounds and can it show growth with these toxic compounds. So here you can see the compounds like 4-methoxybenzoate, ferulate, 2-hydroxyphenyl acetate. These are monoaromatic toxic compounds. And also we used the succinate as a reference compound. Succinate is not toxic compound, but we use it as a reference compound for growth studies. So here you can see the blue line is a growth of Comomonas testosterone with succinate, and the other lines like green, violet are with different toxic compounds. Surprisingly, bacteria showed higher growth with different toxic compounds than succinate. But it took some time, like after 24 hours only, the bacteria was able to utilize these toxic compounds. But in the case of succinate, bacteria utilized it very immediately, like it started from 4th hour and it showed higher growth at 15th hour. But with toxic compounds, bacteria took some time to optimize and to acclimatize its growth. Here we also analyze the concentration of different toxic compounds, how these are degraded by bacteria, and the concentration was analyzed using high pressure liquid chromatography. Initially, we used 3 millimolar concentration of each toxic compound, and all the toxic compounds were degraded completely within 36 hours. But the compound is like 2-hydroxyphenyl acetate was completely degraded within 26 hours. In the left down, you can see the PHB production using 4-methoxybenzoate. Actually, among these compounds, we selected one compound, 4-methoxybenzoate, and then we analyzed the PHB production capacity of Comomonas testosterone. So this 4-methoxybenzoate was used at different concentrations like 1 millimolar, 3 millimolar, 5 millimolar, 8, 11, and also we used acetate as a reference substrate. Bacteria produce good PHB with acetate because it is a non-toxic compound, and also it produced PHB with 4-methoxybenzoate. Among various concentrations, bacteria showed good production at 5 millimolar concentration. And after that, we extracted the polymer from bacteria and purified using the methanol precipitation method. So here you can see the extracted polymer, and during cultivation we also collected the bacterial samples at different time intervals like 24 hours, 36 hours, and then we analyzed the PHB granules accumulated inside the bacterial cells. So here you can see these granules. This was done using transmission electron microscope. And in another experiment, we used another bacteria like Spingobium, Chungbikense, and here also we used different toxic compounds like 4-methoxybenzoate, 4-hydroxybenzoate, ferulate, isovalerate. Actually, these all toxic compounds are main degradation products of lignin. But these compounds we can purchase from companies, so those are commercially available. So here you can see the growth of various toxic compounds using Spingobium. The bacteria showed good growth with all the toxic compounds, and also we analyzed the concentration of these toxic compounds using high pressure liquid chromatography. So within 24 hours, bacteria degraded almost all the toxic compounds. So 100% removal was observed except ferulate. The ferulate, it's 90% removal was observed with ferulate. Also, we analyzed the PHB production using different compounds at 5 millimolar concentration. We used 4-methoxybenzoic acid, ferreterate, ferulate, isovalerate, and 4-hydroxybenzoic acid. Among various toxic compounds, bacteria showed high production with 4-methoxybenzoic acid. It was about 60% dry cell weight. Also, here you can see the microscopic image of PHB granules accumulated inside the bacteria. So with this slide, I would like to conclude my presentation. And also, I would like to acknowledge the funding agencies for providing funding for my research studies. First of all, I would like to acknowledge CSIR India for providing doctoral scholarship, and also for my postdoctoral studies, I got funding from different countries like NRF from Korea, JSPS from Japan, and also Humboldt Foundation from Germany. So thank you for your attention. You can ask me if you have any questions, or you can give me some suggestions how I can improve my research further. Thank you for your
Thank you so much, sir.
attention.
Thank you for your valuable words, sir. Yes, we have some questions from our participants. The
Okay.
first question from Presenter Dev is, since bioplastics are made up of organic substances, whether this will interfere with the longevity of the project?
Sorry, can you repeat?
Since bioplastics are made up of organic substances,
Yeah.
whether this will interfere with the longevity of the product.
Longevity of the
Product.
I'm sorry, I didn't get what is longevity. What do you mean exactly?
Um,
It
maybe
means
the
sustainability, or you're talking about?
sustainability. Yes, sir. Probably.
Yeah, because we cannot compare these bioplastics with conventional plastics like polyethylene or polyvinyl chloride, because those conventional plastics have high strength compared with these bioplastics. But we can add some special additives in the bioplastics composition. Then we can enhance these bioplastics sustainability. Yes, we can do by adding some additives.
Okay. So the second question is there any effects of these bioplastic bacteria on human being?
No, because we are using non-pathogenic bacteria for the production of this PHP. And especially in Germany, there are very strict regulations. We cannot use pathogenic bacteria in laboratory, and there are safety levels. So only in some hospitals, they have the permission to use pathogenic bacteria. Even in Japan also same. Actually in India also there are some strict regulations to use pathogenic bacteria. But in my studies, I used all non-pathogenic bacteria. Those are environment friendly bacteria.
Okay, sir. One question from Dr. Biswas among the participants.
Yeah.
Why those bacteria grow better on toxic compound than other compounds?
Yeah, if you see, because all bacteria cannot have that capability, but you can see the two bacteria, Comomonas testosterone and also Spingobium chungue can say. The Comomonas testosterone has a special property because it can use a variety of toxic compounds, and also in my studies it used toxic compounds very effectively and showed high growth. But if you feed these toxic compounds to other bacteria like Pseudomonas or Bacillus, it may not show higher growth than non-toxic compounds. Because this Comomonas has some special enzymes and it has some special genes which can degrade these toxic compounds very easily.
Thank you so much, sir. Thank you for your valuable time. Now I request Dr. Soumya Pandit to pay vote of thanks.
Yeah, okay.
Thank you, Dr. Reddy. It was a really nice talk and it was really valuable. So on behalf of our W2B team, myself as convener, we are happy to express the vote of thanks to you for your esteemed talk on bioplastics for the FDP. So your talk definitely motivated all the participants and us to work with more enthusiasm. And I would like to see in the next conference that we'll organize probably during offline session, and it will be really good to have you among us. So again, I would like to thank you from all the way from Germany. You found some time and you delivered talk, so it was really wonderful. Thank you very much.
Yeah, thank you for having me. I really had a good time with all you guys. Yeah, thank you very much and have a nice day.
Thank you, sir.
Bye.
Thank you all the participants. Tomorrow we'll have our last session and we'll resume on the same time at 10:15 a.m. Thank you for your presence.